So our next oral presenter is Dr. Loh Chen Fei, who will be presenting his title regarding in silico selection and characterization of DNA aptamers against Vibrio parahaemolyticus, LuxP, Periplasmic receptor protein. Dr. Loh Chen Fei obtained his PhD in molecular biotechnology from University Putra Malaysia in 2015. He is currently a fellow researcher at the Institute of Systems Biology, UKM. His current research focus in aquatic animal health and diseases. He's also currently working on subunit vaccine and attenuated viral vaccine against norovirus infection in the respective grouper and giant freshwater prawn species. So Secretary is requested to play his video. Thank you.
Good day everyone. First, I would like to thank the organizing committee for this opportunity to share one of our current research in silico selection and characterization of DNA aptamers against Vibrio parahaemolyticus LuxP periplasmic receptor protein. A brief introduction about aptamers. Aptamers are functional oligonucleic acids that bind to specific target with high affinity. They have been shown to demonstrate unique advantages as chemical antibodies for many applications in biomedicine and biotechnology, as they are non-immunogenic and possess high penetration rate. And they can be developed against unlimited targets. In terms of aptamers production, the aptamer molecule is thermostable and it has less batch-to-batch variation because of the molecules are sequence-specific. And most importantly, the production of aptamer molecules is fast and cost-effective. So in this study, we try to develop single-stranded DNA aptamer as the potential prophylactic or therapeutic agent against the early mortality syndrome that has been affecting the shrimp aquaculture across several countries. The disease is also known as acute hepatopancreatic necrosis disease, in short AHPND. According to the World Organization for Animal Health, the Manual of Diagnostic Tests for Aquatic Animals, there is no applicable vaccination, chemotherapy, or blocking agent against AHPND. The diseased shrimp presented clinical observations that include pale and atrophied hepatopancreas and empty stomach and gut. Vibrio parahaemolyticus has been identified as the culprit of AHPND, where the infection could cause up to 100% mortality. The secretion of soluble toxins and also the involvement of type 3 and type 6 secretion systems are known to be related to the pathogenicity of this Vibrio. So downregulation of the virulence gene's expression in the AHPND causing Vibrio parahaemolyticus is proposed as the mitigation strategies to control the disease. And this strategy is feasible due to the fact that the pathogenic behaviors of the bacteria is coordinated by quarent sensing, which is a bacteria cell-to-cell communication cascade that involves chemical signaling molecule known as the autoinducer. The quarent sensing cascade is cell density dependent, where the autoinducer concentration increase at high cell density and subsequently leads to the expression of genes that are regulated by the quarent sensing. So our target is the LuxP receptor protein, which is a periplasmic protein that involved in sensing the autoinducer 2 in this Vibrio species. Upon sensing the autoinducer 2, the quarent sensing cascade undergoes a series of dephosphorylation leading to the expression of LuxR as the master regulator that controls the pathogenicity of the bacteria. So through in silico approach, we establish an aptamer library and perform molecular docking against the LuxP receptor protein using Autodoc Vina. And we have successfully shortlisted 6 aptamer candidates that possess lowest binding energy. And subsequently, the aptamer protein complex was further analyzed through molecular dynamic simulation using GROMACS to examine the protein and the protein aptamer complexes stability. And then we calculated the binding free energy using molecular mechanics Poisson-Boltzmann surface area approach. All 6 complexes shown different fluctuation profile across 50 nanoseconds of the simulation time, but the RMSD is within the acceptable stable range. And we also observed different numbers of hydrogen bond formation across the simulation time for each of the complexes. And among these 6 complexes, VPL900 recorded the lowest binding free energy that was calculated using MMPBSA approach, but all other candidates recorded binding energy lower than minus 250 kJ per mole. Next, we used the isothermal titration calorimetry to verify the in silico analysis. The nano-ITC is able to record sudden heat changes upon the protein-aptamer interaction. So in order to conduct ITC assay, recombinant LuxP receptor protein of Vibrio parahaemolyticus was obtained by cloning the gene into Pad28B plasmid. And this plasmid was then transformed into E. coli BL21 expression host for the recombinant protein expression. The expressed recombinant LuxP receptor protein was then purified from the E. coli host, and the purified recombinant LuxP receptor protein is approximately 41 kilodalton in molecular weight as visualized in the jar. And then peptide sequencing confirmed the purified protein as the autoinducer 2 binding periplasmic protein, which is the LuxP of Vibrio parahaemolyticus, where the alignment of the sequence peptides shown 76.4% coverage of the LuxP reference sequence. Then prior to ITC, we have modified the aptamer candidates by adding GC complement sequence at the 5' and 3' ends to form a short hairpin structured aptamer. Out of the 6 candidates, only VPL130 and VPL300 showed a significant binding affinity where the dissociation constants were recorded at 0.5 micromolar for VPL130 and 0.2 micromolar for VPL300. However, a decreased entropy is recorded for both the aptamer candidates, and this may be dominantly caused by the decreased conformational entropy in the bound state. This is because differences in protein conformational fluctuation between the free protein and ligand bound states would contribute to the entropy of ligand binding. It is known that the LuxP receptor protein will undergo major conformational changes upon ligand binding, as you can see from the protein structure comparing between the holo and apo structure of the LuxP. And lastly, our preliminary assay showed that upon treatment of the VPL130 and VPL300 aptamer candidates, a significant reduction in bioluminescence of the model organism is recorded, in which the bioluminescence is regulated through the current sensing cascade. The reduction in relative light unit upon treatment of the aptamer candidates showed that the anti-current sensing activity of these aptamers is dose-dependent. So in conclusion, 2 potential anti-current sensing aptamers have been selected through in silico modeling approach to target LuxP receptor protein. And preliminary in vitro assay has demonstrated significant reduction in bioluminescence of Vibrio upon aptamer treatment in a dose-dependent manner. And we are currently examining the anti-current sensing activity of these aptamers in suppressing the targeted virulence genes expression. And this study is supported by the Ministry of Science, Technology and Innovation, and in collaboration with UPM, which the study is funded by the International Development Research Centre Canada. And I would also like to acknowledge the research team for their contributions to the study. And with that, I end my presentation here. Thank you.
Thank you, Dr. Loh. And Dr. Loh is here today to answer any questions from everyone. I don't see any questions in the chat box. Okay, there is a one. Is there a direct evidence in the involvement of T3SS and T6SS in appen pathogenicity?
Hello? Hello, Dr. Sandra, can you hear me?
Yeah, we can hear
Yeah.
you, Dr. Loh.
The direct evidence for the involvement of the type 3 and type 6 secretion system, I think it has been reported in previous study because this secretion system are involved in the secretion of the toxin from the Vibrio itself to the host. Yeah.
Okay. How do you apply this in the farm level, Dr. Loh? How do you apply the Aptamer?
Currently, our research is still at the laboratory scale, which we are trying at the in vitro, through several in vitro study. So we also perform some the anti-QS activity by treating the culture, the bacteria culture with the Aptamers, and then we perform the transcriptome analysis to study the gene expression level first. So in order for us to apply the Aptamer in the farm later on, that is actually another challenge to our research team because we need to modify the Aptamer so that we can stabilize it through the feed. We might incorporate the Aptamers in the feed and then to deliver to the animal, or we can actually apply it in the water. So in any way, we still need to modify the molecule so that it will stabilize in the environment later on. Yeah, we haven't reached that stage yet.
Okay, just one more question, because can the Aptamer has to be like synthesized chemically, or can you find it in natural source of plants or herbs or something like that?
Not really in natural source. Probably we can synthesize it because the synthesis is actually not costing too much. And then no worry about chemical synthesis because the molecule itself is organic, so it probably won't be toxic anyway. Yeah.
Okay, there's no more questions in the chat box. Okay, thank you so much, Dr. Loh.
No
Thank you so
problem.
much
Thank
for
you.
your talk.
Thanks. Yeah.